Welcome to the Biotech Primer BioBasics section, Immunotherapies, Biotech Therapeutic Warehouse, Therapeutic Powerhouse. In this section, we'll differentiate between polyclonal and monoclonal antibodies. We'll describe how therapeutic antibodies are produced. We'll describe the different mechanisms of action employed by monoclonal antibody therapies. We'll give an overview of antibody-drug conjugates. We'll describe bispecific antibodies and their clinical applications. We'll explain immune checkpoint inhibitor therapies, summarize chimeric antigen receptor T cells, and describe liquid biopsies that are used to monitor the progress of cancer treatments. First, though, we'll explain monoclonal and polyclonal antibodies. So in the immunology section, in the basic immunology section, we said that B cells produce antibodies that recognize unique epitopes. So what this means is that there can be multiple different B cells that each produce antibodies that recognize the same antigen, but different epitopes or different portions of that antigen. So here we have 5 unique B cells, each producing a unique antibody, each recognizing a different epitope on that same antigen. And so if I have a tube of antibodies that is a polyclonal antibody, that means that that tube of antibodies contains a mixture of different antibodies that recognize different epitopes. In contrast, again, these are produced by different B cells recognizing different epitopes. In contrast, if I have a vial of antibodies labeled monoclonal, this means it contains one type of antibody. Every single antibody in this tube will recognize the exact same epitope. All right, and so just a word on the terminology here. This derives from monoclonal means one B cell and clones of that B cell, so descendants of that B cell. So mono, one, we have one type of antibody. Polyclonal, poly means many, many different types of antibody recognizing different epitopes. So just briefly, polyclonal antibodies can be used for both research and diagnostic applications. In these applications, we essentially just want to identify, is the antigen present or no? And so polyclonal antibodies are fine for that. For therapeutic applications, though, we must use monoclonal antibodies. We want to make sure that we have a consistent therapeutic effect, so that the therapeutic effect that we see in clinical trials is going to be the same therapeutic effect that we see 5 years later, 10 years later, throughout the lifetime of the product. And we can only guarantee that if the antibody is interacting with the exact same epitope. So that'll give us consistency. And so one of the ways that we can make antibodies is by engineering mice to create human antibodies. And so, originally back in the 80s, when people first tried making therapeutic antibodies, they would just purify an antigen. So for example, the HER2 receptor protein, Herceptin is an antibody that targets that receptor. So we might have first tried to make Herceptin by just injecting the HER2 receptor into mice, purifying B cells from those mice until we found the B cell that was producing the antibody that recognized and silenced that HER2 receptor. But what researchers found when they did that is if you take a mouse antibody and inject it into a human, the human patient is going to recognize that as a foreign protein, and it's going to mount an immune response against that protein. So this is called actually a human anti-mouse antibody response. And so our therapy won't be effective if we have this immune response against it. And so this led to the development of genetically engineered mice, where using CRISPR or other types of gene silencing techniques, we can actually knock out or silence that mouse antibody gene, splice in a human antibody gene. And so now, if we inject this mouse, this genetically engineered mouse, with the HER2 receptor or any other antigen, the antibodies that that mouse produces will actually be human proteins, again, because up here we used a human antibody gene. And so now I can purify this antibody, inject it into human patients, and not get an immune response against the drug. Before I can do that, though, I first have to identify the correct B cell. So just starting from step one, I'm going to inject my antigen, the thing I want to make antibodies against, into that genetically engineered mouse. I'll then isolate B cells from that mouse, and I'll screen each B cell and until I identify the B cell that's interacting with my antigen. And I'm actually going to do additional tests to make sure that it's interacting with the antigen in the way I want it to interact. Is it blocking the receptor? We don't want it to activate the receptor, so we have to make sure that it's blocking the receptor, not activating it. Once I've screened enough B cells, and again, a big part of this is luck. This could take me weeks. It could take me months. If I'm really unlucky, it could take me as long as a year. When I've finally identified that one B cell that's producing the antibodies that I think could be therapeutically effective, I'm going to take the antibody gene out of that B cell, transfer it into CHO cells, Chinese hamster ovary cells, which will be used to actually manufacture that protein for therapeutic use. So antibodies have become, over the past 20 years, antibodies have become and remain one of the most popular types of therapeutics in use today. And a big reason for that is their selectivity. So we really made a point in the immunology section of talking about how one antibody recognizes one specific epitope on its target. And so that means as therapeutics, they're highly selective. And so this sort of illustrates that, this slide illustrates that. We see here our antibody therapeutic only interacting with a specific epitope on its target receptor. In contrast, our small molecule drug is interacting with all of these different receptors. And so the bottom line is that the more specific your drug is, the more target specific, the more selective it is, the more likely you are to have adverse reactions. So a lot of adverse reactions from drugs come from off-target interactions. We're interacting with our target, but we're also interacting with these other things and causing problems. Should be noted that many small molecule drugs also have high levels of specificity. This slide is just making a point. It's probably a bit exaggerated, but the point is that in general, antibodies are much more specific than small molecule drugs and therefore tend to have fewer adverse reactions. They've also proven to be very versatile. We'll look at a couple of examples here in this section, but antibodies have been very successful at treating a whole range of different cancers, different autoimmune disease, cholesterol, infectious diseases. So they're very versatile as therapeutics. And so how do they work? Well, if you'll recall from our immunology section, antibodies carry out specific functions in our body naturally, the antibodies that we produce in response to an infection. And so therapeutic antibodies just essentially do the exact same thing as naturally produced antibodies do, only they do them against an antigen that we've chosen. So for example, Herceptin recognizes the HER2 positive receptor on the surface of breast cancer cells. Rituxan is a lymphoma drug that recognizes a protein that's on the surface of blood cancer cells. And so what happens is when these therapeutic antibodies bind to those receptors, they activate an immune response. They activate white blood cells, such as macrophages and killer T cells, to come along and destroy that cell. And so this also brings up the point that antibodies often work by more than one mechanism of action. So Herceptin, we've already discussed, blocks the growth signal from reaching the breast cell, the breast cancer cell. Herceptin also works by activating an immune response against that tumor cell. Another interesting example is Rituxan. Rituxan, as I just mentioned, was originally approved to treat lymphoma by activating an immune response against those lymphoma cells. Scientists later realized that one of the things, so lymphoma is a cancer of B cells, the patient's producing too many B cells. Scientists later realized that rheumatoid arthritis, an autoimmune disease, is caused by too many B cells that recognize the synovial tissue in our joints. And so they have this idea, well, could Rituxan also be used to treat these patients by reducing the total number of B cells and control their symptoms that way? And in fact, clinical trials were conducted in this second patient population, and the drug was proven to be successful in those patients as well. Another key mechanism by which antibodies work, again, typically in cancer, as cancer therapies, cancer is out of control cell growth, often triggered by having too much of a growth factor receptor present on the surface of the cell, having an overactive growth factor receptor, for example. So if we can identify an antibody that binds that receptor, blocks the signal from getting through, we can stop the growth of those tumor cells. Antibodies can also stop cell signaling simply by capturing the signaling molecule before it ever reaches the receptor, as shown here. And so two examples of drugs that do that are Avastin and Humira. And so what Avastin does, it captures a growth factor that's actually secreted by tumor cells. So the growth factor that cancer cells secrete is called VEGF, vascular endothelial cell growth factor. And what this does is it causes the blood vessels in the surrounding tissue to grow directly into the tumor. And this allows the tumor cell to survive. It allows a direct route for nutrients and oxygen to reach that tumor cell. Without that, the tumor cell is going to starve to death because it's not going to have those nutrients. And so Avastin soaks up that VEGF and stops the supply of food to the tumor. Humira works. Again, it's approved for a range of different autoimmune and inflammatory disorders. If you'll recall, in the immunology section, we talked and we said that a lot of these disorders are caused because the patient is producing an overabundance of inflammatory cytokines. And so one of the main inflammatory cytokines that drives these disorders is TNF alpha. And that is what Humira blocks. So Humira grabs on to TNF alpha, prevents it from reaching its target on a target receptor on the surface of white blood cells, prevents those white blood cells from being activated, and helps to sort of calm down, reduce that inflammatory response. Monoclonal antibodies can also neutralize pathogens. So Synagis is a drug that prevents respiratory syncytial virus in premature infants. So this is a respiratory virus that most babies get, but don't necessarily become critically ill with it. However, premature infants or infants with other underlying health conditions. And this can be life-threatening for those children. And so they are prophylactically given Synagis, which binds the surface protein on respiratory syncytial virus that blocks the interaction of that protein with the receptor on lung cells. ZMapp is an antibody that does a similar thing that blocks Ebola from entering its target cells, which are cells that line blood vessels. And ZMapp is currently in development, has not yet on the market. An exciting innovation that's occurred in the past few years used for antibodies is using them to deliver toxic drugs. And so these are what we refer to as antibody drug conjugates, sometimes just called ADC. And so what this means is we're going to take an antibody that has a specificity for a particular receptor. We're going to attach a highly toxic drug to the end of that receptor, to the end of that antibody. We're also going to choose a receptor that, and this is true for many different receptors, as part of their natural life cycle, it gets taken up or sort of reabsorbed back into the cell. So what's going to happen then is our antibody with the toxic drug attached is going to bind that receptor. Over the course of a few days, it's going to be taken back up, reabsorbed by the cell. And once inside the cell, there's a pH change, so that toxic drug is released, killing the tumor cells. So again, the technical challenges in developing these types of drugs were developing that attachment, that conjugation of the toxic drug to the antibody. It needs to be attached tightly enough so that it stays on the antibody as it's moving through the bloodstream, but then it needs to be able to be quickly released once it's inside the cell. So there's currently, I think, only 2 on the market now. Seattle Genetics and Genentech both have an approved antibody drug conjugate, but there's dozens more in development. People are very excited about these jobs because they allow us to deliver highly toxic chemotherapeutic agents. So chemotherapy can be very effective at killing cancer, but less effective because it is less effective because there's a limit to how toxic we can make the chemotherapy because it has this systemic toxicity. So this ability to use antibodies to restrict the drug only to the target cells means we can use a more toxic agent and be more effective at targeting the cancer. Finally, PCSK9 is a new type of cholesterol inhibitor that was first came onto the market a few years ago. And just this slide first requires a little bit of background on what PCSK9 is. So PCSK9 is a protein that binds LDL receptors on the surface of liver cells. And the normal function of LDL receptors is to absorb excess LDL, that's low density lipoprotein cholesterol, which is the bad type of cholesterol, the type of cholesterol that is associated with heart disease. Normally, the LDL receptor is present on the surface of liver cells and helps the liver reabsorb excess cholesterol so it doesn't damage blood vessels and cause heart problems. The PCSK9 molecule, when it binds those receptors, it causes them to degrade, so it makes them disappear. So higher levels of PCSK9 protein lead to decreased LDL receptors and higher LDL cholesterol in the blood. And so 2 different products currently on the market work to inhibit that PCSK9 protein, so prevent it from reaching its target here on the surface of the liver cells. So this in clinical trials, this was shown to reduce cholesterol levels, LDL cholesterol levels, as much as statins. However, it has a better safety profile. So there's about 10 to 25% of patients that take statins have serious side effects that make them not able to take the drug. And so these drugs would be a solution for those patients. And finally, bispecific antibody constructs, a new type of antibody. So most antibodies that exist in nature that are sort of naturally produced by our immune system, most antibody therapeutics that are currently on the market, both arms of the antibody recognize the same target. So Herceptin, both arms recognize that HER2 receptor, and so on. What a bispecific antibody is an antibody that has been engineered using genetic engineering techniques in the lab. Scientists can splice, cut and dice DNA from two different proteins. So essentially they can take the DNA coding for one arm of an antibody, paste it together with the DNA encoding a second antibody arm, and in so doing, the protein, the antibody that this gene makes, one arm will recognize, for example, a cancer cell, the other a T cell. And so what happens is this brings that T cell in close proximity to the cancer cell, and it activates the T cell to directly attack that cancer cell. Again, only one, I believe, has been approved by the FDA, but many are in development. People are very excited about this new type of therapy as well. Immune system checkpoint therapies is another type of immunotherapy that's been getting a lot of attention over the past several years. So first, a little bit of background on what an immune system checkpoint is. So the immune system, as we've described, is a very powerful tool. So very strong way to get rid of foreign invaders. But we also have to be careful not to activate it against our own body, or not to activate it when it's not necessary. And so our cells, our immune system cells, have evolved these things called checkpoints, so that if they start to attack our own tissue, this checkpoint will shut it down, will make it so it doesn't attack our own tissue. And so an example of that is this PD-1 protein on the surface of T cells. When PD-1 protein is activated, it sends an inhibitory signal to the T cell, shutting it down, telling it not to attack. And so one of the things, or the thing that will activate PD-1, is a protein called PD-L1 on the surface of all of our healthy tissues. So for example, if I had a killer T cell that accidentally tried to start attacking my liver cell has these PD-L1 proteins on the surface. They would engage the PD-1 and say, stop attacking. I belong to you. I am self tissue. Shut it down. So this is great. This is one of the things that protects us from immune responses. Cancer cells, though, not all cancer cells, but certain types of tumors overexpress. So they make extra copies of that PD-L1. And so they've done this so that they will really quickly shut down any T cell response that is initiated against them. So T cells, eventually, once the cancer is mutated to a certain level, T cells will recognize it as foreign and something that needs to be gotten rid of. But if we have extra copies of PD-L1, that shuts down the T cell. So a checkpoint inhibitor, so these PD-1 proteins are called checkpoints. So a checkpoint inhibitor is an antibody that either binds PD-L1 or it can bind PD-1. And either way, it's going to prevent that interaction. And so that T cell will remain active and able to attack the tumor cell. And so again, a number of these have been approved and second generation checkpoint inhibitors targeting still other checkpoints are on the market, are currently in development. So CAR T therapy, another very exciting development within cancer immunotherapies. And the CAR T stands for chimeric antigen receptor T cells. On the next slide, we'll explain in a little bit more detail exactly what that sort of mouthful means. For right now, though, let's just look at the big picture. And that CAR T therapy is simply T cells that have been taken out of the patient's body and genetically engineered to target cancer cells. So these T cells removed from the patient's body. Gene therapy is used to deliver. So an adenovirus, a viral vector, is used to deliver a gene to these patient T cells that will cause them to express a receptor that will make them target or seek out that patient's T cells. And so some of you, I'm sure, are familiar with this. Then know that these have basically been able to cure patients with blood cancers that were not responding to any type of treatment. And we are, for this type of therapy, we are talking about a 100%, it's a cure. It's not life extending. This absolutely wipes out the cancer. So they've been incredibly successful. What the chimeric antigen receptor is the targeting region, so the variable region of an antibody spliced onto the activation domain of a T cell receptor. And the reason for this is because, remember how we talked about how T cells are normally activated by antigen presentation. So it's much easier to target these T cells if we can just use a surface protein. So for example, the CAR Ts that have been approved target a protein on the surface of B cells and treat leukemia, treat this blood cancer. And it's much easier to target than targeting via antigen presentation because we can develop an antibody against that surface protein, splice it onto the T cell receptor activation domains, and those just include domains that are going to, one, keep the T cell alive. So the T cell is only stays alive if it recognizes a target and has a reason to survive, basically. A proliferation, that not only is the T cell going to stay alive, but it's going to proliferate. So it's going to multiply, make copies of itself. It's going to produce cytokines. So it's going to produce some of those inflammatory cytokines that rev up the immune response. And finally, it's going to be cytotoxic. It's going to do what it's sort of designed to do. It's going to inject toxic chemicals into the target that this antibody domain directs it to. So that's where the background on the name chimeric antigen receptor. So I just mentioned how wildly successful these therapies have been at controlling or curing, I should say, previously incurable blood cancers. But the side effects can be very serious, even life-threatening. Essentially, these therapies work so well, they activate such a strong immune response that in some cases of patients have essentially gone into anaphylactic shock. And so now the second generation of CAR T is aimed at developing ways to better control that response. And so one of the approaches being used is a bispecific CAR. So the CAR T now has an activating receptor. So this will recognize an antigen that is present on the tumor cell. And one of the problems is it's difficult to find an antigen that's only present on the tumor cell. So some of the most serious safety concerns have come from CAR Ts being activated to target tissue that was not tumor tissue. And so to address that, these bispecific CARs also include an inhibitory receptor. So we want to identify it's an antigen that is not present on the tumor cells, but is present on healthy tissue that might also include the tumor antigen. So let's say we have an antigen that's present, antigen A is present on breast cancer, it's also present on lung cells. So let's see if we can find a second protein that is not present on breast cancer cells, but is present on the lung cells. So now if antigen A is engaged, the breast tumor antigen B is not present, this CAR T will go ahead and be fully active. If, however, antigen A is present, but also antigen B, the antigen that is specific to lung cells, but is not present on tumor cells, then this inhibitory receptor is going to be activated and send an inhibiting signal. It's essentially going to shut down the activation of the T cell. So we won't have these CAR Ts attacking healthy tissue. So that's one innovation. Other innovations aimed at controlling CAR T include developing small molecule drugs that are required to activate the CAR T. So we would initially administer the small molecule drug. If the patient starts having a reaction, a bad reaction, we can withdraw the drug and deactivate the cells that way. So that's the next generation CAR T, is developing ways to control that response. And so again, there's been 2 approved by the FDA and several more in clinical development. So finally, in this section, a lot of what we talked about was cancer treatment. And so the last few slides are focusing on liquid biopsies, so ways to monitor treatment. So mostly liquid biopsies now are not going to be used for the initial diagnosis, but they are still very important in monitoring treatment. So once we know the cancer is there, are there ways that we can detect whether or not it's going away? And so the idea is we don't want to have to take the actual cut into the tumor every time because that could be very invasive. There could also be risk involved with doing that. And so the liquid biopsy means, can we detect traces of cancer DNA or even cancer cells themselves in blood, urine, saliva, other bodily fluids, rather than a surgical biopsy. And so the most popular way of doing a liquid biopsy thus far is a cell-free DNA. And so this is just based on the premise that all cells die. And again, even though sort of by definition, cancer cells don't die very often, there are still, anytime you have a tumor, there is some small percentage of those tumor cells that do die and release DNA and isolate. And that DNA then is in a blood sample, or in many cases, also in a urine sample. And so we can collect DNA and use PCR technology or sequencing analysis to see what level of tumor cell DNA is associated with that sample. Same idea here, circulating tumor cells can break free from the tumor and be used to detect the presence of cancer. These are very low levels, though, in a blood sample or urine sample, 1 per billion. And so some of the strategies that have been used to try to detect those are magnetic nanoparticles that will be linked to an antibody that recognizes a particular tumor antigen present on those circulating tumor cells. And then we can use a magnet to pull those out of a solution, or microfluidics. So basically a chip that is etched with channels of different sizes and the tumor cells are larger than normal healthy cells, so we can use those channels to separate out the tumor cells from the healthy cells and count them in that way. Exosomes. This is another area of liquid biopsies. Exosomes are small lipid encased vesicles that are secreted from cells. Again, they're secreted from all cells, not just tumor cells, but we can analyze protein, DNA, RNA. We can identify tumor-specific exosomes based on the surface proteins present, and again, found in all different types of bodily fluids, and use those to monitor if there is cancer still remaining in the body based on the level of exosomes or the amount of exosomes collected. And this is just, again, an interesting little side note here. Exosomes is a new, sort of brand new area of research in terms of therapeutics as well. So different companies are looking at potentially using exosomes to deliver drugs. So we mentioned a lot of times a drug can look great when you're testing it in cells or even in small animals such as mice, but we have a problem delivering it to the right place in the human body. And so one idea is if we use these exosomes, they might do a better job of actually targeting human cells because they will fuse with the membranes better than synthetic lipid nanoparticles, because they actually are the exact same material as human cell membranes. And so this is just an example of some of the drugs where delivery has been a problem that this is being looked into for. Standalone therapeutics, again, this is all in very early stage research, but some early stage sort of preclinical work has shown that in a heart attack mouse model, we can repair cardiac muscle by injecting exosomes secreted by embryonic stem cells. And exosomes secreted by healthy cells near tumor cells can cause cancer cell death. So this might, again, have some therapeutic potential. And same idea here with cancer vaccines. Exosomes could potentially elicit a better immune response than tumor antigens or tumor DNA, which has had mixed success. And so this slide, again, many different liquid biopsies actually already on the market. So in this section, we talked about a very hot area within biotech today. We explained what a monoclonal antibody is, how those are made. We talked about the different mechanisms therapeutic antibodies work by triggering an immune response, blocking receptors, capturing signaling molecules, capturing pathogens, and increasingly by delivering a toxic drug directly to tumor cells. Genetic engineering has been used to create a new class of antibodies called bispecific antibodies, and these can recognize more than one target simultaneously. So, for example, they can be used to bring a white blood cell, such as a killer T cell, in close proximity with a tumor cell. Antibodies are increasingly being used to deliver toxic drugs in the form of antibody drug conjugates. Immune system checkpoint therapies enable the immune system, specifically T cells, to become fully activated even when a cancer cell is trying to shut them down. CAR T therapies engineer T cells to target a patient's tumor cells, and liquid biopsies offer a non-invasive alternative to standard biopsies for monitoring how all of these great new treatments are working in a patient. So thank you. That concludes our section on immunotherapies.